season, and you can see on the website in the coming years. Our first speakers of the fourth season is Dr. Augusto Villanueva, who is currently associate professor at the Icahn School of Medicine in Mount Sinai in New York City. He received medical training from University of Santiago de Compostela in Spain, and he's a board certified in gastroenterology and hepatology in Spain. Prior to his current position, he was a senior lecturer at the King's College Hospital in London. Currently, he combines clinical activity as a hepatologist with research focused on understanding the role of intratumor heterogeneity in cancer evolution and the development of novel minimally invasive biomarkers using liquid biopsy. Then he had published more than 140 publications, many of which published in high-impact journals such as the New England Journal of Medicine, Nature Genetics, Gastroenterology, Hepatology, Journal Hepatology, etc. He has served as an associate editor for the Journal Hepatology and Liver Cancer. He's currently the executive secretary of the International Liver Cancer Association. Now, without further ado, let's welcome Augusto. Augusto is all yours.
Thank you very much. Let me share my presentation. Can you see the slides?
Yes.
Excellent. So first of all, I would like to thank Xinwei and Anu and Tim the opportunity for giving this lecture. We've known each other for many years, and it's a privilege to give a lecture. And the NIH has been one of the major forces to understand genomics in HCC, and I've admired your work for many, many years. So here I have a list of my disclosures. Nothing is really relevant except for the last one, the patent of a signature that we'll discuss at the end of my presentation. And this is the outline with the three topics that we'll be discussing today. First, I would like to frame the type of clinical problems that I'm trying to address with the research that we do in the lab. And then we'll dig into tumor evolution heterogeneity and the work that we've done in that area. And finally, we'll talk about liquid biopsy and why I think is going to be, a major player in biomarker development for HCC and other malignancies, particularly in two clinical aspects: early detection and prediction of treatment response. So just to give you an overview of the impact of liver cancer worldwide, you have here a plot that shows the estimated number of incident cases for different tumor types and the estimated number of deaths. Liver, you have it here as the fourth. This is the data from 2018. Is the third. The fourth depends on how it compares with the stomach cancer. The size of the dot that you see here is the ratio between new cases and deaths, which provides kind of an estimate of the lethality of the tumor. So liver cancer is the second most lethal malignancy right after pancreatic cancer. So you have a tumor that is frequent, that is lethal, and that is increasing. For instance, here in the US, between 2009 and 2013, you have here the different tumor types, the incidence of the different tumors, and despite liver and intrahepatic bile duct cancer are not among the most incident, when you see the delta, the variation between 2009 and 2013, a little bit dated, but what it shows is for both men and women, liver cancer is the tumor type that has increased the most in terms of incidence in that period of time. So again, an increasing problem in the US and worldwide. The natural history of HCC is very rare to develop HCC if you have a healthy liver. You need chronic damage, chronic inflammation, degeneration, mostly related to viral hepatitis, alcohol use disorder, or non-alcoholic fatty liver. Develop chronic hepatitis and cirrhosis, and that stage, cirrhosis, is the tipping point where the risk of HCC significantly increases. The estimate is between 2 and 3% per year. So out of 100 cirrhotic patients, every year you have 2 or 3 developing HCC. This is very variable depending on the etiology and other clinical factors, but is what is used to recommend surveillance in these patients. And we'll talk a little bit more about surveillance and the importance. There are some patients that can develop HCC in the absence of cirrhosis. Some patients with hepatitis B, because we know the virus can integrate in the host genome and can trigger some oncogenic events. Patients with NAFLD can also have HCC before cirrhosis, and similarly, patients with hepatitis C and F3 advanced fibrosis, not to the stage of cirrhosis. In any case, the problem with these patients is that since they do not have cirrhosis, they're generally not enrolled in surveillance programs. So when the tumors are detected, they tend to be at advanced stages when there's no curative options. Now, for the cirrhotic patients, what is recommended today is ultrasound plus minus serum alpha-fetoprotein every 6 months. And Amit Singhal did a meta-analysis a couple of years ago that showed that with that approach, you get a sensitivity of 63% to detect early stage HCC. So you could argue that, with 40%, almost 40% of the early stage cases not being detected, the tool is not, a fabulous tool. I have to note also that, so the first mention of AFP associated with HCC, I found in this Lancet paper from 1970. So it's been more than 50 years of knowing this association, and despite all the efforts, we still don't have a minimally invasive biomarker that can help us in the early detection of HCC. In my opinion, clinical unmet need. Now let's talk about singularities of HCC. As you know, you can diagnose HCC without the need of a tissue biopsy. And the reason for that is the behavior of HCC on contrast-enhanced imaging, MRI or CT scan. You have here on the right an example of a CT scan of a patient with cirrhosis. The patient has ascites. There are a number of nodules that are hypervascular in the arterial phase. They wash out in the portal venous phase. That is enough to call that an HCC and treat the patient accordingly. Now, this is very convenient for patients because they don't have to do a biopsy, but it's a missed opportunity in terms of access to tissue for biomarker studies. And that has been an endemic problem in HCC. And I have to say that there are some attempts now from the high, at the high level in terms of guidance to change that and not force, but, recommend biopsies as a standard of care for patients, regardless of the imaging criteria. There's another very interesting thing about HCC, and I found in this science paper published, again, a number of years ago. So they analyzed the RNA-seq data, so gene expression data from 10,000 samples that were deposited on TCGA, again, different tumor types. You have here a very simple PCA plot that shows relative similarity of the different samples in the gene expression space. Each dot represents each of the samples, and they're colored or based on the origin, the tumor type. And what you see here that the principal component 1 is essentially separating roughly the different cell of origin. So lineage dominates or has significant impact in that principal component, meaning that that's what captures more difference between the samples. But then the second principal component analysis is essentially these purple samples that are very different to the rest. Well, actually, those are the HCC samples. When you group all the samples in a single dot, you have all the tumor types, gene expression, again, roughly grouping together. And then you have the HCC that is right here on the top right corner, roughly similar to glioblastoma. So when you look at gene expression, there's something fundamental that makes it different in HCC compared to the other tumor types. And the authors argue about, the metabolic component of the liver and how this could impact, different gene expression profiles that is so different to the rest. But still, there's something clearly different in HCC compared to the other tumors, at least from a gene expression perspective. There's another difference in this when you look at actionable or druggable mutations. So the folks at Memorial analyzed 10,000 tissue samples from different tumor types, including 100 HCCs. They analyzed 500 genes for mutations, the MSK impact panel. And they did something that I found very clever, is that they ranked the mutation based on levels of druggability, let's call it that way. So how likely is that a specific mutation being targeted by a FDA-approved drug, or there's a biomarker that identifies this mutation as responsive to a specific medication? And they rank the different tumor types based on the highest level of druggability. You have GIST as number 1, as expected, but HCC is second to last. So only mesothelioma has less druggable mutations than HCC. And that's a problem. We're talking about targeted therapies, but that has changed more recently. And one of the last pieces that I want to mention that I found it very interesting conceptually is that, so you know that in 2008, the results of the seminal SHARP trial were reported where sorafenib for the first time, a systemic agent, was able to significantly increase survival in patients with advanced stage HCC, with a remarkable effect size, I would say, in terms of hazard ratio. Now, people thought, well, if sorafenib works in advanced stages, it makes total sense to test it at earlier stages. So it was tested in combination with TACE, different trials. It was testing as adjuvant therapy after resection. All of these trials were negative. So you could argue that despite the same disease and the same treatment that is effective in advanced stages, for whatever reason, is not effective at earlier stages. So as you progress in the disease in terms of the stages, it's not only that you have more disease, more tumors or larger tumors, is that, again, there may be something fundamentally different in the disease compared to earlier stages. And think from a molecular monitoring perspective, that's very interesting. And the final key introductory point that I want to make is, so the last 5 years has been a revolution in the management of HCC from the point of view of systemic therapies. And just 2 weeks ago in ESMO, we have 3 clinical trials being reported, 2 positive and 1 negative. So the options for systemic therapies have changed significantly. So everybody's scrambling to say, which is the best sequence of treatments that would maximize clinical benefit and minimize toxicity. And they're using, the clinical trials, the information from the trials, the group analysis from the trials, the clinical characteristics of the patients, and it's reasonable because what we have. But I argue that, well, there may be a more, reasonable way to do that, and it's using biomarkers that will tell you, based on the characteristics of the tumor, which is the best therapy for that specific patient. And we know that among the different evolutionary constraints that can change the trajectory of tumors, treatment is one of the strongest. So when you treat a patient, you get a response. When the tumor comes back, there are some changes in the clonal composition of that tumor. So we somehow need to have information of what's the genomic or genetic makeup of the tumor at different time points during the treatment journey of the patient. And that cannot be done with tissue biopsy, in my opinion. We need something else that can be readily available, and that's where liquid biopsy enters the space. So in summary, I would say that the deaths due to HCC are increasing worldwide. The WHO estimates there are going to be 1 million deaths by 2030 in the world. Early detection tools are suboptimal, sensitivity is 63%. Most cases are still diagnosed in intermediate and advanced stages. We have limited access to tissue biopsies. Still, the non-invasive criteria for HCC is widely applied in clinical practice. We don't have biomarkers except AFP for ramucirumab, but it's kind of a silo in the broader spectrum of systemic therapies of HCC. So there's no biomarkers that could predict response to systemic therapies. And the application of sequential multi-regional tissue sampling to understand the patterns of evolution after therapies is unrealistic. So we need something else. Otherwise, we'll miss that critical information to understand mechanisms of resistance in HCC. So with that background, let's dig in the issue of tumor heterogeneity and how I think it links with evolution and resistance to therapies. So we and others, including Xinwei and Anu, have worked extensively on molecular classification of HCC. And the concept is very simple. Tumors are at the same clinical stage, they're different molecularly. And there are different ways that you can categorize that. We've used gene expression signatures that correlate with outcome, but you can throw also DNA methylation, epigenetic data, you can have microRNA data. So there are ways, and it's been well established, that you can classify or understand the different subclasses of HCC when you look at genomic data. And that has implications, at least in terms of outcome, less so in terms of prediction of response for a number of reasons. And one of them is the limited access to tissue. There's no question that there is a way to categorize tumors using molecular data. Now, when you do the exercise of looking, for instance, at HCCs when they're relatively small, this is not very common. what I'm going to explain, but it happens. When you ask the pathologist, you know, you had this an HCC, can you describe in more detail the type, you know, the degree of differentiation, the immune infiltration? In this case, and again, it's a little bit extreme, you have a 1.3 centimeter HCC with 3 different areas that are histologically different in terms of degree of differentiation or tumor grade. Poorly differentiated, meaning more aggressive, and well differentiated, meaning less aggressive in this tiny tumor. Well, that's not very common, but it does happen. And similarly, it happens with immune infiltrate. There are areas where you have scarce immune infiltrate within the same tumor nodule, as opposed to others where the infiltration is more heavy, more intense in the same tumor nodule. So the question is, well, depending when you do the biopsy, you may get different information. I'm talking about morphology, but what about genetic data or molecular data or genomic data? Is that so? Let me give you a more practical example. So this is a resection specimen from an HCC. Here in yellow, you have the tumor. If we assume that the diameter of this is 3 centimeters, the volume of the theoretical spherical tumor is 14 cc's. If the size is 5, the volume is 65 cc's. But if the size is 15 centimeters, and you may think that it's very big, but patients with advanced stage may have this amount of tumor, the volume is 1700 cc's. So the volume of a biopsy from a 14-gene needle is approximately 0.16 cc's of this cylinder, which represents 1% of the 3 centimeter, 0.2% of the 5 centimeter tumor, and 0.00009% of the 15 centimeter HCC. And the key question is, so with 0.00009% of the whole tumor mass, are you able to recapitulate the complete landscape of genomic alterations present in the tumor? And the answer is, it depends on how homogeneous or heterogeneous tumors are. Because if they're very homogeneous, it doesn't matter. They're going to be the same all over the place. So we try to address that question. And to do that, we selected 14 patients, treatment naive, early stage HCC, all of them treatment with surgical resection, where we did geographic sampling of the tumors and the adjacent non-tumoral tissue. As you can see here, there are different sizes of the tumor, and you can see where we actually took the samples and the regions that were, you know, selected for further analysis. And I have to say that we try to avoid areas of necrosis or areas of hemorrhage to make sure that we're capturing actual cells. We did bulk genomics, RNA-seq, targeted DNA sequencing, and copy number alterations. In total, were 72 biopsies from 12 patients. 6 of the patients had hepatitis B virus infection, and this is relevant for a thing that we'll explain in a minute. And in these 2 patients in green, we did single-cell RNA-seq. So in each of these areas, we did single-cell with the 10x chromium, roughly 6,000 cells per region, but with a much more density in terms of gene expression at the single cell level. So this is the first piece of information. You have here, again, a very rough portrait of similarity of the different regions. PCR component analysis, the circle, so each data point represents a region that was analyzed with RNA-seq. This is gene expression space. The circles represent tumor regions, and the triangles represent non-tumoral adjacent regions, and they're colored by the patient of origin. As you can see, there's a very nice demarcation between tumors and adjacent non-tumoral tissue. And for the majority of cases of tumors, all of the tumor regions for the same patient, they co-cluster together. So you have all the regions of patient 10 are here, all the regions of patient 8 are here, of patient 6. There are some patients where at least one of the regions could cluster closer to another region of another patient than to the other regions of the same tumor nodule. For instance, look at patient 2. You have here 4 regions of patient 2 in blue, and the 5th region is down here. Or patient 11, 2 regions for patient 11, one is here, the other is here. So there's something that makes these region outliers in terms of gene expression that makes it really different. So why is doing that? Well, we're doing RNA-seq data, and you get reads from the tumor cells and from the tumor microenvironment, and we can scale the degree of outlier with the amount of, let's say, tumor cell composition. So the lower the amount of tumor cell composition, in other words, the higher the amount of microenvironment component, the more heterogeneous the sample is, which makes sense. And in our case, even though we have stroma, the immune infiltrate was a major driver when you look at the gene expression data. So we got interested at that interface between the immune cells and the tumor cells. So we use the RNA-seq data to quantify expressed mutations. So this is the algorithm that we use. It's well established, is computational, so it's prone to errors and inaccuracies, but it's the best approach to estimate not only the expressed mutations, but the predicted immunogenicity of those mutations. So here what we're showing is that for region A of patient 10, tumoral region, you have a density plot of all the expressed mutations that we detected. In the y-axis, you have the binary fraction of that specific mutation. So here you have subclonal mutations, opposed to here I have more clonal expressed mutations. And here you have the binding affinity, the predicted binding affinity of that expressed mutation, of that neopeptide, neoantigen, predicted again. The lower the binding affinity, the higher the immunogenicity of that specific mutation. So the more likely of that mutation to elicit an immune response. So here's region A, and I'm going to show you the rest of the regions of patient 10. And they're very similar, and we can quantify that computationally. So it's very similar. There's not dramatic shifts in terms of the distribution of the neopeptides. This is the patient that I just showed you, patient 10. I'm going to show you patient 2. And here we do see dramatic differences. So the distribution of neopeptides is different according to the different regions. So B, C, and D, they're relatively similar, but A and E, they're very different. And this tracks with the degree of immune infiltrate. So we cannot prove that there's a causal association, obviously, but there is at least an association between the intensity of immune infiltration, I'm talking about T-cell immune infiltration, and how the clonal composition in terms of express mutation is being shaped. Let's move to the single cell data. Remember, we had 2 patients. We did RNA-seq in different regions of the tumor. We have here again a resection specimen. Here you have the tumor delineated. Again, we try to avoid the areas of necrosis and hemorrhage. These are the 3 regions that we did a single cell RNA-seq. What you have here is a Disney plot. Each point represents each of the cells in this analysis, and they're colored based on the region that they were derived. So in green, region A, here, region in purple or pink, region C, and in yellow, region B. Now you can tell the computer to tell you which are the marker genes that differentiate different clusters. And when you look at the different marker genes, you see that what you're essentially identifying as suspected is lineages. So you have HCC cells, you have B cells, cancer-associated fibroblasts, endothelial cells, and myelin-derived cells. So when you recolor that Disney plot with the lineage information, you get the majority as suspected at HCCs, and then you have the cancer-associated fibroblasts, the B cells, endothelial cells, etc. Now in the second patient, patient 14, we had 4 regions sampled. This is the Disney plot. Again, it's colored by the region. When we compare, and I know it's only 2 cases, but you compare it with the patient 13, the Disney plot is much more sparse. The data is less confluent. We took these marker genes the same. We didn't select new ones. We took these ones, applied them here, and we again identified the same cell population. So HCCs, CAFs, B cells, etc. But in this case, there was a new population of cell here in yellow. Based on the markers, we knew that were cytotoxic, but we don't know if they were T cells or NKT cells. And that's where we leave it as cytotoxicity NKT cells. And again, it's only an N of 1, but one could argue that the reason why this tumor is much more sparse, there's more difference, and when you look at the molecular classification at the cell level, you can see that there are different classes of molecular classes within that tumor, could be related to the fact that there's more cytolytic activity, confirming or suggesting that, again, the immune system is a major constrictor in at least at early stages of tumor evolution in HCC. Now, we got very interested in hepatitis B, and this is a paper from a group in Spain that analyzed the pattern of integration of hepatitis B virus in HCC cells. And what they found, they timed the prediction or the estimation of when those events were happening, the first initial relevant integration of hepatitis B, like TERT promoter or CCNE1, etc. And I found very interesting when you think about evolution and how, the perspective of time in clinical practice, how early on and we're talking about years, those events were happening. Since we were doing poly-A bridge poly-A tail RNA sequencing, we can also identify expression of hepatitis B genes, because as you know, hepatitis B, the mini chromosome, uses the machinery of the cell to express its genes and its products. So you can somehow quantify the level of expression of hepatitis B in our samples. Here you have the 10 samples. Remember I told you there were 6 from hepatitis B patients. You have here 1, 2, 3, 4, 5, and 10. The ones that we did not detect any hepatitis B because these patients were not infected with hepatitis B. You have here the different levels, the quantification of the expression of hepatitis B in these, for instance, in patient 1, in green you have the non-adjacent tumoral tissue, and in red you have the tumoral tissue. And one thing that we found, at least I found very interesting, that we didn't follow up, is that, for instance, in patient 1 and patient 3, when you look at the tumor and the adjacent non-tumoral tissue, we did not detect any expression of hepatitis B in the regions of the tumor, but we detect significant expression of hepatitis B in the adjacent non-tumoral tissue, suggesting that somehow the tumor cells have developed, I wonder if as an evolution mechanism, very sophisticated ways to shut down the expression of hepatitis B virus. Maybe not very interesting from a cancer perspective, but very extremely interesting from a virological perspective. We haven't followed up on that. There are other studies that show this heterogeneous expression of hepatitis B in tumor cells, and I think it's very interesting. At the end of the day, what you have is that, as we know, tumors are complex ecosystems that have tumor cells, but other components, immune cells, cancer-associated fibroblasts. In the case of HCC viruses, that is very relevant for a number of reasons. So for instance, I didn't show the data, but you can compare directly the immunogenicity of the antigens from the virus and the antigens from the tumor. And the antigens from the tumor are proportionally much more immunogenic, predicted immunogenic, than the antigens from the virus. So you have 2 potential sources of immunogenicity in a tumor, and I think it's very telling of the complexity of HCC. So I want to move to the last part of my presentation that will deal with the role of liquid biopsy in the clinical management of HCC. And we'll go back to the concept of early detection and the natural history of HCC. I mentioned that, you know, there are some patients that can develop HCC before the stage of cirrhosis, but let's stick to the 2-3% of the cirrhotic patients that will develop HCC every year. That's the target population of surveillance. If we put some specific numbers to that, there are approximately 600,000 cirrhotics in the US, and this is again an underestimation, most likely. We have 2-3% per year that will develop an HCC, so you have 18,000 new HCCs every year. There are more than that, but let's stick to that number. We assume that the sensitivity of ultrasound and AFP for the detection of early stage HCC is 63%. We detect roughly 11,000 HCCs, so we have 6,500 patients that are not detected because sonogram and AFP are not good enough to detect these tumors. Well, this is the ideal scenario, but it's far from reality, because we need to compute an additional factor here, is that how many of the patients with cirrhosis are actually enrolled in a surveillance program by which they do every 6 months a sonogram and an AFP. Well, the reality is that only 24% of the patients are actually in surveillance, which is a very low number, and we can talk about the reasons for that. So if we go again back to the 18,000 new HCCs that are developed every year, and we consider that only 24% of the cases are actually in surveillance, when we apply the performance of the ultrasound and AFP, we see that we only detect 15% of the total number of new HCCs, with a staggering 85% that are not detected either because the patient is not in surveillance or because the tool is not good enough. So there you have the 2 main barriers for early HCC detection. The first one is we need a better tool. So ultrasound and AFP is just not good enough, at least based on the meta-analysis. This is variable depending on where you do the study and who does the ultrasound, but let's stick it to the global data. And the second thing is that we need to increase implementation. So why only 25% of patients with cirrhosis are on surveillance? If you think about it, just by increasing that number, so 25% to 50%, using still ultrasound and AFP, the number of new patients that will be detected with early stage will increase dramatically. So both factors, I think they're somehow connected when you consider that one of the main reasons why only 25% of the patients with cirrhosis are on regular surveillance is attrition. So the patient, they go to the sonogram and AFP, they have to travel mostly in rural areas. They do the surveillance and everything's fine. They go 6 months later, everything's fine. They go 6 months later, everything's fine. So they get a little bit tired. So why I'm doing this? It's a waste of time. It's too much. I'm not interested. Everything will be fine. So you lose a lot of patients in that situation. So I wonder if there's a way to simplify both things or to address both things, meaning that can we improve the accuracy of the tool and at the same time simplify the whole process of HCC surveillance so that patients don't get tired and they continue to be engaged with HCC surveillance. And that's where the concept of liquid biopsy comes into place. I want to mention this paper from the group of Xinwei Wan on the power of viral exposome of VARScan, the technology that developed to estimate or to use it as an early detection tool. Again, a non-invasive tool that is outperforming AFP for the early detection of HCC in different cohorts. And that's the concept. So can we use something in the blood that doesn't need to do an ultrasound? The patient doesn't need to travel. It's very simple. You can collect the blood in your local CVS or in your local LabCorp. You can send it to a central lab and you get a prediction of your risk of HCC. And that's essentially the concept of liquid biopsy. Liquid biopsy is the analysis of tumor components, either cells, ctDNA, or vesicles that are released to the bloodstream. It can be isolated and analyzed, and that correlates with relevant clinical events such as tumor development, treatment response, outcomes, etc. Now, when you think about liquid biopsy, there are many different analytes that you can choose. And I mentioned these three main groups, CTCs, exosomes, ctDNA. By far, the most exploded and the most advanced in terms of clinical development is ctDNA. There are others. ctDNA, these are fragments of DNA that are released to the bloodstream when the tumor cells die, either by necrosis or apoptosis. And you can look at different things in ctDNA. You can look at mutations of the ctDNA. ctDNA circulating tumor DNA. You can look at epigenetic marks, and we'll talk about that. You can look at deletions, amplifications, and we'll look at fragmentomics, and we'll also talk about that a little bit. The other aspect that I think is interesting is circulating tumor cells. When you look at the data of CTCs, until relatively recently, people were focusing on counting the amount of CTCs. And it's tricky because we're talking about literally one CTC among a million of white blood cells. So it's very challenging technologically to make sure that you're isolating CTCs. Now, more recently, the effort has shifted a little bit to, instead of just counting the number of CTCs, and this correlates with different clinical outcomes, is to actually isolate the cells and do genomic molecular analysis on those cells. It's like a truly liquid biopsy. You're like getting similar information to a tissue biopsy in terms of the breadth and depth of information from mutation analysis, gene expression alterations, but you're using the CTCs. And that's very appealing, but again, it's challenging. And also, so the ability of tumors to release CTCs can be very variable. I also want to mention a very recent study from the guys at Stanford that show that the analysis of the fragments of ctDNA can help infer gene expression. So a lot of people are not concerned, but they didn't like about ctDNA is what is very DNA-centered, very focused on changes in DNA, and you don't get such a breadth of information then of gene expression that we know is very relevant. So what they show, they developed this metric, the promoter fragmentation entropy, that correlates with the length of the fragment of DNA, and that, you know, somehow scales with the expression, the transcription side, sorry, and that that metric correlates with the actual gene expression they did in lung cancer. No, the higher the metric, the higher the expression for a number of genes that are known to be upregulated in lung cancer, and the genes that are downregulated were also the metric was low. So my point being that the amount of information is increasing. You not only have access to mutation analysis or copy number alterations or epigenetic alterations, now you can estimate, and this is a first step towards understanding gene expression using fragments of ctDNA in the blood. And the Holy Grail, and there's a company called Grail that is addressing this, is can we use that as a general cancer screening tool? So every year, most of you go to your PCPs and you do blood work to check for your cholesterol and your A1C, how's the diabetes, do we have risk factors for cardiovascular disease because of the cholesterol, triglycerides, etc. The concept is you would do something similar to check for cancer, the cancer screening tool. And these guys, it's a study from Johns Hopkins, a very comprehensive study, a very provocative study that tried to address that in 10,000 women, and they were specifically targeting ovarian cancer. As you know, majority of patients with ovarian cancer are detected at advanced stages when the patients have already ascites and survival is challenging. So I want to see, can we, using this technology, and CancerSeq is a combination of known tumor markers plus ctDNA markers, can we use that for the detection of cancer when they see the performance, even though the negative predictive value was very high, when you look at the when you look at the sensitivity of the different combination, either blood only or imaging, it was relatively low, 30%. That's not scalable clinical, that's not useful clinical, but it is a proof of principle towards the idea that there would be eventually a more sensitive way to detect cancer as a general screening platform on a population level. And that's where the field is going, I think. Now, going back to HCC, it's very attractive to think that, well, if we can use liquid biopsy for the early detection of HCC, we don't need to do the sonogram, as I mentioned before. So these are two studies that address that, and they use specifically DNA methylation alteration, which is in the circulating tumor DNA. They're, they follow up with companies and they're developing advanced studies, but in their initial studies, this is a study from China, 1,000 HCCs, 800 controls, a number of methylation marks in ctDNA with a sensitivity of 85%. Remember that ultrasound AFP was 63. Now, 85% in the training, 83% in the validation, specificity higher than 90%. It doesn't get better than that. Again, there's some issues with the study, and we'll talk about that. Second study in the US, very similar approach. Again, a number of methylation marks, different to the ones from the Chinese study. Very high sensitivity for patients at early stages. Now, these two studies have the same problem. The main problem is that the majority of patients that were enrolled did not have early stage HCC. And you don't need a tool to detect tumors when we're talking about surveillance. You need a tool to detect early stage tumors, because otherwise you may not improve survival. And the objective of surveillance is decrease cancer-related mortality. So that's why it's so important to include early stage tumors. And the performance may be different when you do early and advanced stage. We got interested in vesicles as a source of potential biomarkers in liquid biopsy, because at that time when we started the project, there were emerging information about the complexity of the cargo that these vesicles have. And there were a number of papers that suggested they were functional. So it's a kind of a sophisticated mechanism of communication between cells, which they can change the phenotype. And there was a very interesting paper suggesting it could have a role in priming metastases from pancreatic cancer in the liver. So we got interested in extracellular vesicles. And we did a very simple study, a very pilot study, where we took plasma from patients and controls with HCC, early stage HCC, and we did ultracentrifugation to isolate the vesicles. We did a number of tests to make sure that we were isolating vesicles, including immunolabeling, NTA, electron microscopy, and Western blot. And then we did RNA-seq on these vesicles, on the content of the vesicles. And we found what we call a small RNA cluster. So these, for instance, you have here this, what we call PILAB. So a number of reads, a long, tail of reads that map to a specific area. There were others who we can assign a level of complexity of that small RNA cluster in terms of the distance between the clusters that we found, and I emphasize this is within the extracellular vesicles. And we focus on these with low complexity. And we found that no matter the method that we use to isolate the vesicles, either using ultracentrifugation or nano-DLD, which is a method patented by IBM to isolate vesicles, the correlation of expression was very nice. And also when we analyze the origin or the specific location of the expression of these small RNA clusters, they were specific to the vesicle. So the expression differ when you look here in terms of the cellular component and the vesicle component. So the cells are actively packing the small RNA clusters in these cells. What do they do? We don't know. Maybe probably, or just garbage, cellular garbage, but somehow we thought that it could be used as a biomarker. And the reason for that, because in this pilot study where we have 15 patients, 10 HCCs here in red, and 5 controls, we analyzed the 250 more differentially expressed small RNA clusters. As you can see, it's very easily to discriminate between tumors and controls. We selected the top 3, the top higher in terms of complexity, lower complexity, and we expanded the analysis to a larger cohort. Here we have 15 patients, we expanded to 190 patients. Approximately 100 had HCC and 90 were controls without HCC, but enrolled in surveillance programs. So these are the patients at risk of developing HCC. And when we analyzed the expression of these 3 small RNA clusters, they were significantly overexpressed in patients compared with tumors. And again, I want to emphasize that all of the patients had early stage HCC. So when you look at performance of our 3 small RNA clusters in the vesicles, here you have the predicted performance of ultrasound and AFP. Again, sensitivity is 63%, specificity is around 83%. And the first line here in blue is the performance of AFP in our cohort, which is quite nice and quite good. And we think that the reason for that is because, there are less hepatitis, untreated hepatitis C, and the AFP may perform better in this cohort. The second line here in orange is the performance of our 3 small RNA clusters, so better than AFP alone. But the one in the top is a combination of AFP plus the 3 markers. And, we think that AFP has still a role in the early detection of HCC. But what's interesting is when you compare the expression of the 3 markers and AFP, there was no correlation, meaning that, both components capture complementary information in terms of early detection of HCC. Now, the next step, and if some of you are familiar with extracellular vesicles, the field is very complex because there are different ways of isolating the vesicles, and ultracentrifugation is kind of the most frequently used, but it's not clinically scalable. You cannot do the ultracentrifugation in a clinical setting looking for an easy and practical way of detecting early HCC using blood markers. So we got in contact with Brian Cunningham from University of Chicago, Urbana-Champaign. And he has developed this method, DNA total probe-based photonic resonator absorption microscopy, which essentially prevents the use of amplification, PCR, that we were using in the previous study. And he could use this. I'm not going into details of the technology because it's a little bit cryptic to me, but essentially you were able to very sensitively detect your RNAs of interest. So we apply that with our 3 small RNA clusters. You can see here in these are the small RNA clusters in a control patient and in 4 HCC samples. There is a nice dose relation when you look at different concentrations. The signal that you get is much higher. And as you can see here, the HCC cases for the 3 small RNA clusters have significant higher expression. So there you have a very simple, clinically scalable way to quantify these 3 small RNA clusters that could be applied in clinical practice eventually. So the summary of this part is how do I see early detection in the next 5 to 10 years? Well, we need ways to, among all the patients with chronic liver disease, and you name it, can be cirrhosis, can be NAFLD, can be hepatitis B without cirrhosis, the ones in red that are at higher risk of HCC development. So we need ways to stratify that, to risk stratify these patients, to identify the ones that are truly at high risk that would benefit from early detection or surveillance. And you can use different ways to do it, germline DNA, viral features, degree of liver disease, clinical variables, etc. Now, in these patients, I think the future of HCC surveillance is blood-based. So it will be either ctDNA or fragmentomics or the vesicles that we're developing, but blood-only method that, along with clinical data, clinical variables that are very relevant when you estimate risk of HCC, can give patients an individual probability of the patient having HCC. And that's the information that is actionable. If the patient has a 90% probability of having an HCC, after all this trouble, then is when you can say, okay, we need a confirmatory test in the form of an MRI or a CT scan. What happens at the end of the day is that you're removing ultrasound from the equation, and you have a more accurate way of detecting early stage HCC, and something that is more easily applicable in clinical practice. So the prediction is that you will not have 25% implementation, you will have more implementation rate if you simplify the whole process. Now let's move to the final part of the presentation, biomarkers of treatment response. So we published this, and Giuseppe was the first author of this sort of algorithm, and it's already outdated. And the reason for that is because, as I mentioned last week, there are 3 new trials reported, 2 of them positive, 1 negative, but essentially there are 2 first-line options for the management of HCC at advanced stages in terms of systemic therapies, atezolizumab or tremelimumab, durvalumab, and we can argue which is the best way or who should be used first. And they have the second-line therapies, could be sorafenib or lenvatinib, if you cannot apply any of the 2. And they have the third-line therapies, which are the known effective tyrosine kinase inhibitors that were approved based on randomized data against placebo. So it's also a little bit dated, but it's the data that is available, like sorafenib, cabozantinib, etc. And they have single-agent immune checkpoint inhibitors, or the combination of nivo-ipi, which is approved under the accelerated program. Again, a rather busy set of options for the management of patients with early stage or with advanced HCC systemic therapy. So how can we put a little bit of order here? So one of the things that is key in this whole thing, in my opinion, is that when you look at the atezolizumab plus bevacizumab data versus sorafenib, positive trial, breakthrough, hazard ratio very strong, 0.58, new standard for first line. When you look at the objective response rate, and here's the data from the paper, it's been updated to 30%. So 30%, 1 out of 3 patients have an objective response rate. And those are the patients that drag the clinical benefit. So in my opinion, the key question is, who are these patients? So who are the ones that do so well, that drag the outcomes, and they have long-term survival? And bear in mind that duration of response is one of the key factors that prompted the FDA to approve single-agent immune checkpoint inhibitors in HCC. So it's not not only the amount of responses, but the duration of the responses. So they do very well. This is a study published very recently that addressed this issue for atezolizumab. Andres is the first author. You have here, they took different cohorts that were exposed to atezolizumab plus bevacizumab, the trial that I just mentioned here, the IMbrave 150 and other clinical trials. They applied different type of analysis, gene expression, genetic profile, etc. It's a very dense paper. I don't want to get into much details, but there's truly some signals, right, of potential efficacy when you compare, for instance, PFS or overall survival in the ratio of Treg and T effector cells. There are differences with opinion of interaction, favoring response to atezolizumab as opposed to non-response when the ratio is high as opposed when it's low. And I think that's interesting is the CTNNB1 mutation. So this is a gene that is part of the Wnt signaling pathway that is mutated in roughly 30% of the patients with HCC. And what is interesting is that not a long time ago, but 10 years ago, there was some data, and we work on that, that suggests that sorafenib could target the Wnt beta-catenin pathway. It's interesting that in patients with mutation of beta-catenin, the benefit of atezolizumab is lost compared to sorafenib, again suggesting that sorafenib may somehow target the Wnt signaling pathway. So we just published, and this was led by Giuseppe with Philip Harper as the first author, a gene expression analysis on potential prediction of response to checkpoint inhibitors. Again, this is pre-atezolizumab, so I wouldn't say that it's dated, but certainly the field is changing very rapidly. You have here the different cohorts in first line, in second line, and a signature that is associated with response to immune checkpoint inhibitors. You can see here in terms of overall survival and PFS, you have here the specific marker genes that define the signature, as expected, a lot of genes related to immune response. And you can see the performance compared to other signatures that have been reported for prediction of response to immune checkpoint inhibitors in other malignancies. So again, the AUC was maximized with the signature. And the final thing that I want to mention is that, so there was a debate 5 years ago that is still ongoing about what's the impact of beta-catenin mutations in response to immune checkpoint inhibitors. You know that there's, again, 30% of the patients with mutations beta-catenin, and those patients, some of them have immune exclusion, even though not all of them. And there's this concept that was, released by the guys' memorial that patients with beta-catenin mutations, they did not respond to immune checkpoint inhibitors. In a very tiny study of 27 patients with HCC, you have here the curves. Again, there were no differences in patients treated with sorafenib, but the ones we have mutations in Wnt signaling, they do very poorly with immune checkpoint inhibitors. This was kind of a rumbling noise that you have beta-catenin mutation, you're not going to do well with immune checkpoint inhibitors. And Maya here at Sinai provided some, mechanistic suggestion of why this was happening, and she developed animal models expressing it with expression of an antigen, glucose specifically, in different genetic backgrounds, and show similar results that when you have expression of, so when you express beta-catenin, the background of immunogenic host, of albumina, the response was not significantly different. So we tried to address that, but the problem is, I mentioned before, we don't have tissue biopsy for the majority of patients that are treated with immune checkpoint inhibitors. So we use liquid biopsy. We did ctDNA mutation analysis of 85 patients with advanced HCC. We selected a very small panel of genes, 25 genes targeting beta-catenin, the most common suspects. And as expected, first thing that we found is that, the usual suspects in HCC, p53, beta-catenin, they are mutated at, I would say, relatively similar frequencies, and they're mutually exclusive. But the key thing that we found that was very interesting is that we did not validate this finding that, if you have beta-catenin mutations in the blood, and there were bona fide mutations, that you did not respond to checkpoint inhibitors. We didn't find that. There was a similar response between both of them. Actually, we have one patient with a beta-catenin mutation that had a fantastic response to immune checkpoint inhibitors. I'm not saying that the previous hypothesis is discarded completely, but there's some nuances on subclassification of the beta-catenin subclass in terms of its relation with response to checkpoint inhibitors. We also played with the concept of sequential sampling, and we have some patients that we have blood at different time points of the evolution of the disease. 2 patients, 7 and 8, that we have blood from pre-treatment. Patient had a complete response, durable complete response. Then the tumor came back, and you can estimate the clonal composition of the blood that could suggest potential mechanism of resistance. In one case, in one patient, we had an emergence of a p53 mutation that was not detected at baseline. Again, rudimentary, but a kind of a proof of concept that, in my opinion, that's the way to go in terms of molecular monitoring, and make it in a way that is realistic. And we have work also in animal models to develop that. The final 2 things that are 2 ideas I want to give you is how so one is academia, then is the industry in terms of the contribution to liquid biopsy. So currently, the industry ecosystem is estimated with a market share of $2 billion. And you have here examples of some of the players that are addressing, not only early detection of HCC, but early detection of cancer screening and analysis of minimal residual disease. There's a projection that this market size in 10 years will be roughly 20 billion, so a 10 times increase in the market size of liquid biopsy products related to cancer detection. And the final piece of information is the FDA is also on board. There's a guidance that just released this year on the use of ctDNA for early stage solid tumor drug development. It's a guidance from industry. So again, it's clear that we're at the first stages of this technology being implemented. So finally, I will conclude that despite major advances, HCC has become a major health problem based on the incidence and mortality that I think is still key to understand the mechanism of resistance, and understanding the tumor heterogeneity is a very appropriate way to understand the mechanisms of resistance. And ideally, if you could predict how tumors will evolve, will be ideal, such as we do in viral diseases. I think liquid biopsy has emerged as a very promising and convenient tool for biomarker development. I think early detection is for sure the number one niche that I think it would be the first application of this technology, but also prediction of treatment response. And that molecular monitoring, defined as a real-time tracking of molecular alteration in HCC, I think will improve early detection and treatment allocation as long as we have the tools to implement it safely. And finally, I want to thank, I forgot the thank slide, but essentially there's a number of people that worked in these different studies. Johan von Felden was a postdoc that worked on the extracellular vesicles, and also Amanda Craig, that is a postdoc now in the lab. It was critical for a number of studies, and particularly for the intertomal heterogeneity study. And thank you for your attention. I'm happy to take questions.
Great. Well, thank you so much, Augusto. This is actually really a very nice overview in terms of the problem, the challenging, and also some of the example that from your laboratory. So it's very appreciative. So I think that we have a few minutes for questions. I don't see any in the chat box. Perhaps if you have a question, maybe you can raise hand, and so we will get to it.
Augusto, I have one. So, I can understand that you stress the importance of early screening in patients with cirrhosis. But honestly, I think it's actually the wrong approach. Because if you really want to do early screening or early detection, you have to piggyback on what is done in general with other types of cancers as well. And you have to dismiss cirrhosis, because you will increase the acceptance that these tests will be done dramatically if you just piggyback on regular, annual screening methods. Having said that, I have a question for you. You showed this very early science paper where they looked at all the different tumors, and the HCC was so different than all the other cancers other than glioblastoma. Does it make it more difficult if we now put the screening methods for, use the same screening methods that we use for other tumors also for HCC?
So for your first question, so the problem to do it at a population, if I understood correctly, you're suggesting to not go to cirrhosis and do it at a population base.
Yes.
The problem with that is that the risk of HCC on a population base is so low that it's very difficult to have any technology that is performing well in this setting. So we'll have a lot of false positives. So that's why you need to concentrate on the patients at the highest risk, where you make it cost effective to do the surveillance. In terms of the differences between, I still cannot, get my head around that figure and that paper. I don't have an explanation. I mean, the metabolic explanation is partial, but the difference is, to me, dramatic. And I don't know if we can pull the same methods that we apply for other malignancies, but I think we need to still be relatively specific. So the pathways and the alterations are very tumor dependent, and I don't think of a universal way that we can just detect tumor regardless of the tumor site.
Okay, I always admire the very challenging question from Tim. I think this is a very interesting point. Obviously, it's deserve for a debate in terms of a high risk versus the general population. I totally agree with Augusto in terms of the cost effectiveness. But speaking of this part, and I think that so detection of cancer, early detection of cancer versus risk prediction, they are probably two different things. Currently, there are methods already well established, and most recently is the Korean study about Plan B that really survey large populations, and you could really assign individual into low risk, medium risk, high risk, as what your the outline slides about, enrich for 90% of the patient who are likely to develop a liver cancer, and then you intensively screen those people. So what are your thoughts in terms of just simply the risk model that has been developed, but instead of really trying to find those new methods for early detection, risk prediction?
No, I think that I agree, and there is a frequent misunderstanding that people pull together risk stratification with early detection. They're completely different clinical problems, and they need different tools. So I think that The problem with the models that are out there is that they are not very well validated. Their focus, so if you do Asia, is hepatitis B centric, and they're not terribly validated in the West. But for instance, the methods to predict risk in hepatitis B, there's also the issue of the genotype being different of hepatitis B and some sub differences there. So I think that we still need good tools to risk stratify the patients. The clinical information on the models will be good, but I think we can do a little bit better. Thinking about the ones that you would say, your risk is very low, you don't need to do screening. That's a big decision from a clinical perspective. You have to be sure that the patient will not develop HCC at a reasonable pace.
Oh, good, thank you. So Mandy has a question. Mandy, do you want to open the audio video and to ask question?
Hey, hi Augusto, how are you doing?
Great to see you.
So my question was on the biomarkers that I'm always seeing are in retrospective studies mainly, and there are so many proposed biomarkers. And I was wondering what is kind of like the studies that are going forward. Are there prospective studies being done to test those, or that's just not being reported because they don't have positive outcomes? Is that why AFP is still like the only one? We're just like not testing them in the clinical setting well?
Yeah, so the answer is yes. There are a number of studies, prospective studies, phase 2, phase 3 biomarker studies that are ongoing, testing mostly DNA methylation in the ctDNA. That are designed to address that issue in a prospective setting. So the quality of the information that you get is much richer. That's why I think in the next 5 years, we may see a significant change in how early detection is applied, because now you have high quality prospective data on what's the performance of these new technologies for early detection. There are other technologies, the glycotest and other studies. So the answer is yes, and they're relatively advanced, large number of patients, well designed. I think this industry now have the tools that are very competitive, and they want to test it in the appropriate way.
Great, thank you very much. Any other question from the audience? Okay, if no further question, let's give a big round of applause to Augusto. Very nice work.
Thank you very much, guys. It was great to be with you.
Thank you all for attending.
Thanks, you. Bye.
Bye-bye.